Okay. So, welcome everyone to another webinar from the ISCB Student Council. Before we start, a short reminder that you can also submit abstracts to be featured in this webinar. Just also follow the social media announcements. And today I'm very happy we have Dr. Kalpana Raja with us. She's a postdoc at the Morgridge Institute at the University of Wisconsin. And she's an expert at text mining and natural language processing. And today she will be telling us about how to use that to better predict adverse drug events. So Kalpana, the floor is yours.
Thank you. So before I start, I just want to make sure that the screen is visible at your end.
Yes, I think that's all looks fine.
Thank you. Thank you so much for confirming. Hello, everyone. Thank you for giving me an opportunity to share my knowledge today. I'm going to talk about enhancing predictions of adverse drug events using drug-gene interaction. The approach used is text mining. As a traditional approach, we need to read article after article to get all the information. This is tedious, time-consuming, and actually impractical. Text mining, on the other hand, it's an automated approach. It's quick and efficient and accurate. In our experience, we see that it can read 30 million articles in few minutes. So now, let's see how text mining can be done. The main resource for our research is PubMed articles. Currently, PubMed contains more than 30 million articles. Luckily, it is free for use. The first step in text mining is to get the subset of articles which we are interested in. For example, if you are a researcher doing some research in Alzheimer's disease, you will be interested to get the literature related to Alzheimer's disease. So retrieving those literatures is called information retrieval. So once we have the subset of literatures, the other steps in text mining come into picture. So let's see information extraction. That is a common step next. Automatically, we can get the information from PubMed articles, whatever we are interested in. Some of the examples include gene-gene interaction, gene-disease association, gene-chemical interaction, gene-pathway association, and there are many more. So once we have the information, extracted the information from PubMed, what can we do? We can either go ahead and build knowledge bases so that it becomes public and others can use it. Some of the examples include Comparative Toxicogenomics Database and GSEA. Or we can just integrate the informations together for hypothesis generation that finally leads to knowledge discovery. In this way, we can find some knowledge which is not yet published, like predicting something. As I mentioned already, the main resource for our research is PubMed articles. But text mining researchers cannot use the online PubMed database because there are so many people around the world and everybody wants to access the online database. It will be a disaster. So there is a different way to do this. Along with the online version, PubMed also provides XML files for download. So these XML files include the entire online resource. Each XML file includes multiple PubMed articles. All the informations, whatever you see online, are available in this XML files. So whatever is required for our research, we can just retrieve those information. Especially in my research, I'm interested in retrieving the titles, abstract, mesh index, and chemical index. If you want something else, it's possible to get those informations as well. So for retrieving these informations, in text mining, we write some program which we call it as parser. So once we get the information, the next step is to build a personalized PubMed. When we have the personalized PubMed, we have to make sure that the retrieval from the personalized PubMed should be of the same speed as we access the PubMed database online. To achieve this, we normally index and then compress into a personalized PubMed. There are several softwares. The most common one is Apache Lucene. So Apache Lucene indexes and compresses, then it builds the personalized PubMed. The personalized PubMed can be carried in the laptop, around 33 gigabytes in size. So once we have this, we can just query the personalized PubMed for all our text mining research. So the first step is citation retrieval and processing. So as I mentioned earlier, we are not going to deal with all the 30 million articles. We will get a subset. So here I'm going to say how we can get the subset and what we are going to do with that. There is something called eUtilities. So using this, we write the query to get PubMed IDs which we are interested in. eUtilities can get only the PubMed ID, nothing else. So once we have the PubMed ID, again we use Apache Lucene or similar softwares to query personalized PubMed to get abstracts related to the PubMed IDs that we retrieved using eUtilities. Once we have the abstracts, then we have to process. So it's a pre-processing. So we developed something called text processor that breaks the abstracts into sentences, and we assign PubMed ID to each of the sentences so that later on we can know where the sentence comes from. So up to here, it is just retrieving and doing some pre-processing. So we have all the sentences ready for the study. So today, my talk is on drug-drug interactions and adverse drug events. And I'm going to show how we can enhance the predictions using the genes. So the biological concepts involved are genes and the drugs. The first step is to find the gene mentions or the drug names within the sentences. So this is my approach. There are various approaches people use in various ways. This is my approach. There is a resource called Gene to PubMed, and it's from NCBI. For each PubMed, they provide annotated genes. And the resource includes only the gene ID. Since my research is interested and we are interested in humans, we just retrieved the PubMed articles related to human. So when I did this research, there were 498,000 PubMed articles related to human. We all know that all the PubMed articles do not include abstracts. So we restricted PubMed articles with abstracts and annotated with up to 5 genes. So that gave me 469,000 PubMed articles with 20,000 annotated genes. So this is the data. So the next one is gene mapping. So some of the steps, it is just the repetition. So this part, I'm not sure whether you can see my cursor. So if you can see up to this PubMed sentences from the personalized PubMed, we already discussed. Instead of eUtilities, we already have the PubMed articles. So we just follow the same one and we get the PubMed sentences. As I mentioned earlier, the annotation in Gene to PubMed includes only gene ID, but gene IDs do not appear in PubMed abstracts. So we got the aliases, names, and symbols for every gene annotated for every annotated genes from Anthregene. Then use that resource to find the gene mentions within the PubMed sentences. The first step is exact string match. Here we assume that the researchers write the gene aliases, names, and symbols in the same way as it is mentioned in anthragene. Of course, we all know that this is not true always. So we also went one step ahead, which is called normalized match. So here we developed a rule-based system so that we can bring uniformity between what the researchers write in PubMed articles and what is mentioned in anthragene. So finally, we got PubMed sentences mapped with genes. So our approach obtained 73.6% of recall for the exact match, and we were able to increase this recall by about 9% when we combine exact and normalized matching together. So we achieved 82.2% recall. We got several publications only for this gene finding and gene normalization. That's what you see at the bottom of the page. So the next step is mapping the chemicals and drug mentions in PubMed sentences. We developed a chemicals and drugs lexicon from various resources. I will tell in detail in the later on slides. So we use this lexicon to find the drugs and chemical mentions in PubMed sentences, for which we used a tool called MedTagger. This is from Mayo Clinic. So we just used that and we just gave the lexicon to MedTagger. So finally, it gave us PubMed sentences mapped with chemicals and drugs. So one side we map the genes, other side we map the chemicals and drugs. So finally, we filtered the sentences with at least one gene and one chemical or drug. And for drug-drug interaction, we also filtered set of sentences with at least two chemicals or drugs. So let's just recall what is the topic for today. I already said that I'm going to show you how to enhance the predictions of adverse drug events or adverse drug reactions through drug-gene interactions. Adverse drug reactions pose critical public health issues affecting over 6% of hospitalized patients. Knowledge on drug-drug interaction and adverse drug reactions is necessary to give proper treatment to the patients. However, it's very challenging because a lot of drug discovery researches are going on and a lot of papers are getting published every day. So this is our pipeline. As I mentioned earlier, different people do in different ways. So this is our pipeline. So first, we developed the chemicals and drugs lexicon from various resources. We used UMLS Metathesaurus, Drug Bank, and Form GKB. The ID across the resources are different. So we combined them and gave a customized ID to identify the duplicates across the resources. Our approach gave us 1.2 million chemicals and drugs along with the synonyms. Our pipeline includes text mining as well as machine learning for classification. Especially, we want to include the machine learning to get more reliable results, and text mining and machine learning together gives very good results. This I can say from my experience. So we used an annotated corpus called drug-drug interaction corpus. That includes the documents from Drug Bank and Medline. So we used our chemicals and drugs lexicon to find or to map the drugs present in drug-drug interaction corpus. We did this for both training data as well as test data. On the other hand, we have the mapped sentences from PubMed. For drug gene association, we used comparative toxicogenomics database. So we got the drug pass, we got the common genes, and the association types from this resource. So finally, we gave all the resources to the machine learning engine. First, we trained the model using the training data from drug-drug interaction corpus. So machine learning, definitely we have to talk about the features. We use two sets of features. One is related to drug-drug interaction. The second set is related to drug-gene interaction. First, we classified the drug-drug interaction, which means we got the drug-drug information from the text mining, which is like a sentence includes at least 2 drugs. So we try to classify whether it is true or false, just a binary classification. In the next step, we try to find the adverse drug reactions. Again, it is the same set of features. So once we confirm drug-drug interaction, we see whether it can lead to adverse drug reactions. So this is our result. For the drug-drug interaction corpus, we achieved 0.94 F score for the training data and 0.88 F score for the test data. We use the same training model to predict drug-drug interaction and adverse drug reactions on PubMed database. So here I have to report on 2 sets of features, drug-drug interaction feature and drug-drug interaction and drug-gene interaction features together. So for drug-drug interaction features, we achieved 0.80 F score for predicting drug-drug interaction and 0.87 for ADR. When we combine the features together, we were able to increase the F score. We got 0.81 F score for drug-drug interaction and 0.9 for ADR. Thus, we show that by incorporating drug-gene interactions, we can enhance the predictions. We also tried with various classifiers to see whether it is true what we say, like enhancing the predictions using drug-gene interactions. Yes, we achieved the same results. So that's what you see on the right. Drug-drug interactions and drug-gene interactions together achieved better predictions than using drug-drug interaction features alone. We applied this model for drugs for cutaneous diseases. Various diseases we used. Here I'm taking psoriasis as a disease model. So our model successfully retrieved the information present in the literature, as well as it predicted new information that are not available in the literature. So some of the examples are below. So here I'm showing the prediction that has already the literature evidence. That's what I'm showing in the braces, PubMed ID. Methotrexate is a known drug for psoriasis. It has interactions with probenecide and NSAIDs, non-steroid anti-inflammatory drugs. The ADR type is adverse effects. So this is our prediction, and it's already there in the literature, supported by the literature. The second example is the interaction between cholecalcitriol and parathyroid hormone levels. The predicted ADR type is effect at molecular levels. So the ADR types are actually from the DDI corpus. They have 4 types of ADRs. We just adapted whatever is defined in the corpus. So predictions using DDI and DGI features together. This is the best model. So what we predicted. There are 32 drugs for treating psoriasis. 8 drugs show interactions with genes in comparative toxicogenomics database. Among that, 3 drugs were predicted to have drug-drug interactions and adverse drug reactions with 10 other drugs. So the 3 drugs are shown in green circles: cholecalciferol, methotrexate, and mycophenolic acid. They show interaction with 10 other drugs that are shown in yellow color. One of the example is estrogen through intermediate genes. So that's what you see here. Between the two drugs, there is an intermediate gene. As I mentioned earlier, we were able to predict the known information in the literature. So that's what I'm highlighting here. So some of them it's from PubMed and some of them are from UpToDate. We were also able to predict new DDI and ADR information. So I'm highlighting here, it's a lot. So this is the model and this is the prediction. All these new DDIs are not yet published, but we can show the information through the interacting genes. For all these new DDIs, the corresponding ADRs are shown here. As I mentioned earlier, we just adapted the ADR type from DDI corpus. So if you see the first example, methotrexate and antifolates, the gene, interacting gene is DHFR. The adverse drug reaction is adverse effects. We were able to find literature evidences for this one. The next example is the second one, methotrexate and diclofenac. So here I say that unknown as adverse drug reactions. There are only 4 types of classifications for adverse drug reactions in drug-drug interaction corpus, but sometimes we also face that we couldn't accommodate the ADR type into any of them. So we just had a 5th classification for our research where we just say unknown. Yes, ADR exists, but we cannot fit into the available 4 types of adverse drug reactions. So for the PubMed ID, I'm not giving anything, which means the information is not in the PubMed, so it's a new prediction. With this, I'm concluding my talk today. I have a lot of people to acknowledge from my current lab, from my previous lab at University of Michigan, USA, then a list of funding agencies. Finally, I thank the people who selected me and gave me an opportunity to present my work today, and the audiences for your time. Thank you so much.
Okay, thank you very much for this talk. So for the people in the audience, if you have any questions, you can just post them as a comment on YouTube, and then we will, Kapana will answer it. So please do so if you have any questions. In the meantime, could I just clarify what exactly the output of your prediction model is? Is it sort of a categorical thing of, okay, this is the drug-drug interaction? Or do you also get something of a confidence level about the prediction?
Yes, of course. We just calculated some score, so the confidence level. Based on the score, we just had a cutoff. I forgot the cutoff, what cutoff we fixed. So above the cutoff, whatever we got, we say that yes, the drug-drug interaction exists.
Okay, and so, so I was wondering, so you know, that something is found in literature is maybe not immediately also true, sort of, if it's one paper. So I was wondering if you did anything with sort of the level of evidence for, so if you have 10 papers describing it, for example, you can find it.
Yes, we got that level of evidence.
Yeah.
Because especially for the machine learning part, so we just did the matrix. So for everyone, every entry, we just considered all the PubMed articles that include the co-occurrence of drugs or drug gene.
Right. And so I was wondering, as I was listening to this, it seems that this is an area for linked data, where there's a lot of applications for linked data, because as you mentioned that, yeah, okay, there's many aliases for genes. Also, people use certain different words to describe the same thing. Is there here also an effort to maybe create like ontologies for this to make it easier to make it machine interpretable?
Maybe. Though the resources are available already, I do see a lot of errors there. So especially for genes, it's very challenging when we include the synonyms. The same
Yes.
synonym appears across many genes. So
Yes.
when we target that particular synonym in a PubMed article, which gene is going to be that? So
yeah,
that's a question. So we get some false positives.
Yeah,
Right
I can
now,
see that.
I think we can get the information from anthrace gene, and we have to avoid the synonyms that are common to 2 or more genes.
Okay, yeah.
We may miss some articles, but at least we can control the false positives.
Yeah, I guess that is sort of the more important part of it, because otherwise the false positives explode. So yeah, we don't have any questions from the audience, but maybe it is nice to share that Selva Kumar says that he likes to work in drug discovery, and your webinar will help him very much to get to the next level.
Oh, thank you so much. That's a positive feedback. Thank you.
So yes, with that, if there are no questions from the audience, I think that concludes the webinar for today. So thank you very much for joining us. And then we will end it now.
Thank you so much for the opportunity. Thank you.
You're welcome.